Okay, welcome everybody. Hello, it's my pleasure to introduce our speaker today, Dr. Holly Muller from UC Santa Barbara. And I've known Holly's work for a while, and I'm not going to tell you a lot about it because she will do it far better than I will. But some of the things that impress me about her is that she bills herself as a mathematical biologist, yet she owns 2 -80 freezers. Correct?
Yes.
So,
yes. Two functional
and I think
-80 freezers.
those two things together, I actually think exemplify a lot about Holly's work. That she is, well, she plans and is practical. She has 2 -80 freezers in case one dies. So that's one thing. But more that she rides this sort of fence between being a mathematical biologist who knows nothing about any organisms at all, and someone who actually knows something about organisms. And that's not a common thing. I think most of us appreciate. Those of us who are empiricists who know nothing about math can definitely attest to that. I'm always impressed when people are able to ride the fence between those two areas and speak to that. And I think that's kind of what we're going to hear about today.
I hope.
Fair enough
yes.
introduction? All right. So without any further ado, I am going to introduce Holly, and please help me welcome her.
Thank you so much, Jay, for the warm introduction and for shouting out our backup -80. And thank you, Claire, and everyone who's taken time to meet with me and walk me around campus and just have so many fun scientific conversations over the last couple days. It is such a pleasure to be here with you all. So I am joining you today from the University of California, Santa Barbara, which is just a few hundred miles south of here. And UCSB sits on the ancestral and the unceded territories of the Chumash people. And particularly as an ecologist, I find it really important to acknowledge that human history of place, which shapes the function of our modern ecosystems, but of course also, and continues to shape today, but also of course we can learn so many lessons from the stewardship of these indigenous peoples. And we're lucky in Santa Barbara to have a really active community, and our local band of Chumash, pictured here, paddling one of their tomol canoes, their traditional canoes that they would use to voyage to the Channel Islands, who are really engaged in the ecology of our area. And in my work at UC Santa Barbara, I also have the privilege to work with a really fantastic team of collaborators, some of whom are pictured here, some of whom you will meet throughout the talk today. And we're lucky to be funded from a diversity of sources, fingers crossed that many of those still exist, into the future. But what I have in common with all of these folks is that fundamentally at our core, we're community ecologists. So we're interested in interactions between species and how those interactions give rise to the structure and function of the ecosystems that surround and support and sustain our own human lives. And we do a lot of this work in the lab and also on the computer, as Jay alluded to, but also we get to sometimes go out into the field. And when we're out in beautiful ecosystems like the conifer forests in Point Reyes or the coral reefs in Moorea, we're looking at the organisms that we find there, and we're particularly interested in their metabolic roles. So for example, when you're out in an ecosystem, you might encounter a tree, which is an autotroph or a primary producer that's really setting the table for the rest of the food web. And you might encounter heterotrophs, like these damselfish, that are consuming organic matter from other species in order to make a living. And these different forms of metabolism shape the ecological niche that these organisms fill. So in particular, if we think about these organisms in their food web context, we know that the autotrophs, like our trees, our grasses, our phytoplankton, are providing the organic matter through photosynthesis that fuels the rest of the food web, and then our heterotrophic organisms are forming intermediate or even terminal linkages. So it's tempting then as a community ecologist to say, if I can understand an organism's metabolism, I can make some important predictions about the role that that organism plays in its community. Now, our textbooks tell us that predicting metabolism should be straightforward, right? Metabolic capacity is encoded in an organism's DNA, which specifies the types of enzymes and cellular structures that that organism can build, and also the assembly of cells, in the case of multicellular organisms, into the whole organism, in this case a velociraptor, with its emergent heterotrophic metabolism. But as biologists, we know that life doesn't like to follow rules, right? And the same is true for metabolism. So there are actually many organisms that acquire metabolism within their lifetime. Today, when I refer to acquired metabolism, I'm describing metabolic capabilities that rather than being hardwired, rather than being encoded within an organism's DNA, that organism instead gains access to within its lifetime through some type of interaction with another species. And because these organisms are then gaining access to novel metabolic pathways, they can expand upon their metabolic niche with implications for their current ecology and also for their evolutionary trajectories. So to make the concept of acquired metabolism a little bit more concrete, let's think of an example that may be familiar to many of you in this room, corals. So corals are heterotrophic animals like you and I, and therefore they need a source of organic material for growth and reproduction. Now, in many parts of the world's deeper oceans, such as in the Darwin Mounds in the North Atlantic, pictured on the left, corals make a living heterotrophically by filter feeding out organisms, smaller organisms from the water column and digesting them. But if I say the phrase coral reef, probably what comes to mind for most of us is something more like the image on the right. In this case, a coral reef from the Big Island of Hawaii, a hotspot of biodiversity, a place that many of us like to visit. Anybody been to a coral reef and snorkeled or dove? There's no shame in raising your hand, right? But one of the reasons we love visiting these coral reefs, these tropical coral reefs, is because the water clarity is fantastic, right? So you can get underwater and you can see all of that diversity. But that clear water is actually indicative of the fact that there's very little food for corals there. There's not too much to filter feed out. So corals have solved this problem, this resource limitation problem, by acquiring metabolism. So if we were to zoom in on a head of coral and look at an individual coral polyp, you can see in the center of this translucent animal tissue here, these brown-green splotches, and these are symbiotic algae that are living inside of the coral cells, and they are photosynthesizing. And some of the sugars that they make via photosynthesis are then fueling the coral's growth. So by harnessing the power of acquired metabolism, corals are not just surviving, but they're thriving, and they're creating these hotspots of biodiversity in parts of the world's oceans that we would not normally expect to find a filter-feeding heterotrophic animal. So that's an example of an acquisition of metabolism that's taking place at the scale of the whole organism. It's what we would term a metabolic mutualism. In this case, a mutually beneficial interaction that's mediated by the exchange of metabolites. But acquisitions can occur at other scales of biology as well. For example, through horizontal gene transfer, where organisms take up and incorporate pieces of DNA that encode for different forms of metabolic machinery. And then also at the subcellular scale through the retention of functional organelles, which I'll talk more about in the second half of the talk today. And given these different mechanisms, right, by which organisms can acquire metabolism, it's perhaps unsurprising that we find examples of these acquisitions and these extensions of metabolism all throughout the tree of life, including in our own human reliance on our gut microbiome that's helping us digest our pears and our muffins from our snacks outside today. In our research group at UCSB, we're really interested in exploring these acquisitions of metabolism to understand how they affect the contemporary ecology of these organisms by allowing them to expand their niches, and then also how they shape future evolutionary innovations. So I'd like to give you a couple of examples of this, starting at the level of the whole organism, thinking about metabolic mutualisms. Now, unfortunately for me, but I think fortunately for everybody who works with me, we do not work on the putative metabolic mutualism between velociraptors and their gut microbiome, a little toothy for our tastes, but we do work on the equally exciting, and certainly today more ecologically relevant metabolic mutualism between trees and ectomycorrhizal fungi. Okay, so trees, business of trees above ground is photosynthesis. They are harnessing the energy of sunlight to produce organic material, and that organic material is setting the table for other organisms in forested ecosystems. But in order to photosynthesize as efficiently as possible, trees need resources like nutrients and water from the soil. Now it turns out that in temperate forested ecosystems, like the ones just south of us in Point Reyes, the oak woodlands by me in Santa Barbara, and in northern and inland California, these trees are gathering those resources by partnering with below-ground fungi called ectomycorrhizae. And here, this light micrograph shows you a few examples of what those ectomycorrhizal associations look like. So the physical branching structures here are the tree's fine root system. And then the coloration, and you can see some of these thread-like fungal hyphal fibers here, that's the fungi that are wrapping like a glove around the tree's fine roots. And it's there at the interface between plant and fungal tissue that resource exchange is taking place. In particular, the trees are fueling fungal growth via photosynthesis, so some of those photosynthetic sugars are being sent below ground. The fungi are using those sugars to grow through the soil, gather resources, and return those nutrients to the host tree. So through the language of acquired metabolism, then, we can think of the trees as acquiring fungal enzymatic abilities that are allowing these fungi to break down organic matter in the soil that the trees couldn't access for themselves. And then the fungi is effectively getting a big carbon straw, a big sugar straw into the sky. Now, one of the things that has fascinated mycologists and fascinated me for many years is the fact that ectomycorrhizal associations can be incredibly speciose. So if we were to go to Point Reyes, for example, to the Douglas fir forest there and excavate the root system of one of those Doug fir trees, we could find upwards of a couple dozen different species of ectomycorrhizal fungi simultaneously coexisting on one tree's root system. Now, we have a few explanations for this, largely centering around the idea that fungal species diversity is related to functional diversity. So we know that fungi are active in different layers of the soil, they produce different enzymes, they're active in different times of year, and together that leads to a hypothesis that perhaps associating with different types of fungi with different functional capabilities allows host trees to have metabolic flexibility to tolerate different environmental conditions. Now, this is a hypothesis that we can test, or at least test the predictions of, through observation. So if we go a little north of here to Mendocino County, there's a really special site there called the Jug Handle Ecological Staircase, which is noteworthy because it sits in a part of North America's coastline that is actually, over geological timescales, rising with respect to sea level. And so what that means is that when you are standing at the ocean's edge, you're standing on soils that are about 100,000 years old from when they arose out of the ocean. But if you go a couple kilometers inland, you're standing on soils that are closer to 500,000 years in age. Now, as soils age, they tend to lose nutrients. And so as we move inland, we start to be standing on soils that have about a third as much nitrogen and phosphorus. They have a lower pH that results in the formation of an iron hardpan, which is this water impermeable layer below the surface that really also affects the plant's access to water as well as to essential nutrients. Yet, despite this relatively extreme environmental gradient, we can find a single species of tree, bishop pine or Pinus muricata, that's able to grow across all of these steps in this chronosequence. And so we might ask ourselves, well, do the ectomycorrhizal fungi associating with this tree differ across this environmental gradient? And in particular, we might predict that in these low resource older soils, these trees need to partner with fungi that are better able to gather and transport resources over long distances. So to test this hypothesis, we literally go out into the field and dig some holes. And we pick out fungal colonized root tips like the pictures that I showed you previously, and we use sequencing to identify the fungi and therefore the functional traits of the fungi associating with these trees. And we look in particular at functional traits that are related to resource transport. So for example, we look at hydrophobicity, which helps fungi transport water over long distances. And what we find is that if we compare in blue the trees on the resource replete younger soils to in orange the trees on the older resource stressed soils, the relative abundance of root tips that are colonized by fungi that are hydrophobic is higher in these resource-stressed older soils, so more capacity for water transport. We also look at rhizomorph formation, so these are these root-like bundles of fungal fibers that allow nutrient transport over long distances, and we find more rhizomorph production in these older soils as well. And finally, we look at the foraging distance, or the physical distance away from the tree's roots that the fungi are looking to find resources to bring back to their host tree. And whereas in these younger resource replete soils, we find a lot of these fungi just looking in the immediate vicinity within a couple millimeters of the root system, on these older resource stressed soils, more of these fungi are foraging further afield. So what this suggests to us is that by associating with a different fungal community across this gradient, a single species of host tree is able to tolerate a relatively extreme gradient in resource availability. So effectively, acquiring metabolism from different partners is letting this tree switch metabolic hats, if you will, depending on the environment in which it finds itself. Now, this is an example of leveraging acquired metabolism over spatial gradients in environmental conditions, but environments can, of course, vary temporally as well. And this is especially relevant for trees and ectomycorrhizal fungi because these are long-lived organisms. We know trees live for decades. Turns out that their ectomycorrhizal partners below ground are living for decades as well. And during that time, they're going to experience environmental change, right? Annual seasonality, interannual variation, decadal climatic oscillations, what have you. And because the ectomycorrhizal fungi are obligately dependent on their host tree as their source of organic matter to survive, if you are a tree and you are going to need this fungal partner in the future, you have to maintain it today. If you stop that investment, then that fungus is going to be lost from the system. So that has led us to a hypothesis that we call the metabolic bet hedging hypothesis, which argues that trees in temporally variable environments should maintain a more diverse, more speciose portfolio of ectomycorrhizal fungal partners, much like a human stock investor who doesn't know which direction the market is going to go might choose to diversify her investments to have the greatest chance at a high return on investment of her funds. Now, as you can imagine, given the timescales involved, this is a challenging hypothesis to test experimentally. And so this is where we turn to our mathematical models, which can allow us to ask questions that might not be tractable in field or laboratory settings. So work done by Dr. Laura Bogar, who many of you know as a colleague here in the plant biology department, where she's faculty at UC Davis on main campus, by Kristen Howard, an undergraduate researcher in our group, and by Dr. Bethany Stevens, who's currently a Simons postdoctoral fellow, explored this idea of whether temporal variation can promote these metabolic bet hedging strategies using a mathematical model. And the way that their model works is that we imagine a tree that's living in an environment where it has a choice of partnering with two fungi. We'll call them the red fungus and the blue fungus. Now let's imagine, just for the sake of argument, that in this particular environment, the red fungus is the higher quality partner, meaning it gives you the higher return on investment, more nitrogen per unit carbon that you give it. Well, it makes sense that a tree that wants to maximize its growth would preferentially invest in the red fungus and maybe even eliminate the blue fungus from the system as the lower quality partner. Now we can imagine another environment, I'll call it environment B, where the opposite is true and the blue fungus is the higher quality partner. The interesting question comes when we ask what happens when the tree experiences fluctuating environments. So it spends some time in environment A and some time in environment B. What would be the investment strategy in its fungal partners that leads to the highest growth? So we can explore that here by looking at the fraction of time that the tree is spending in each of these environments, from 100% environment A to 100% environment B on the x-axis, and then the growth that the tree achieves, the amount of carbon biomass that it accumulates, as a function of its investment strategy. So first, let's think about a tree that invests only in the red fungus. Unsurprisingly, that tree does great in environment A, but if it's spending all of its time in environment B, where that's the lower quality partner, it actually can't survive. And the opposite is true for a tree that invests solely in the blue fungus. But a tree that has a 50-50 mix of investment strategies, that invests equally in these two fungal partners, is actually able to do quite well in a mixed environment, where it's spending about half of its time in each of the two environmental conditions. Although notice that here in the extreme environments, on the edges, the tree has lower fitness. So this is kind of a canonical bet hedging result, where you are reducing your fitness in the extreme cases to also reduce variance in your fitness, leading to greater performance in variable environments. To me, this is an interesting phenomenon, not just for thinking specifically about tree ectomycorrhizal fungi, but also for what it says to us broadly about the maintenance of mutualisms and diversity in multi-species mutualisms. These are textbooks, right? They talk, when we talk about canonical mutualism theory, we often imagine that mutualisms are under strong selection for partner recognition and sanction mechanisms, because that's how you prevent your mutualism from being overrun by cheaters. But the bet hedging idea argues that there might be some circumstances under which having a more general investment strategy, or maybe making some mistakes that allow you to maintain a more diverse portfolio of partners, results in higher performance. And so these two pressures, selection for specificity and selection for generality, might push back and forth against one another over evolutionary timescales. This is, of course, an interesting theory to put forward based on mathematical models. It's another thing to try to demonstrate that empirically, which we're trying to do now in partnership with Laura, using experimental systems in the lab where we manipulate tree seedlings and their associations with fungi. So more to come, stay tuned on this. And we're also continuing, I can't help but give a shout out to a former undergraduate researcher in our lab who's now also here at Davis. We're also continuing to explore mathematical models of these systems, and in particular, how these different investment strategies and the reliance on partners as your source of nutrients and the reliance on this acquired metabolism can shape how these systems respond to disturbance. So you can ask Jacob all about that at a later time. Okay, so through our study of metabolic mutualism, acquired metabolism at the scale of whole organisms, we can therefore see how these acquisitions allow organisms to extend their phenotypes and enable them potentially to tolerate more variable environmental conditions in both space or time, with, of course, complicated implications for what the evolution of these metabolic associations has looked like historically and will look like into the future. So having spent some time thinking about these acquisitions of metabolism at the scale of the whole organism, I want to shift to thinking about what these acquisitions look like at subcellular scales, particularly through the retention of my most favorite organelle, the chloroplast. So remember, the chloroplast is the eukaryotic organelle for photosynthesis, and our textbooks tell us that some organisms have chloroplasts, like plants and algae, and some organisms don't, like animals, protists, some protists, fungi, do not. But there are organisms out there today that are stealing chloroplasts and becoming transiently photosynthetic. So we call these organisms kleptoplastidic, klepto for stealing, like kleptomaniacs, plasty for the chloroplast. And these organisms literally eat something photosynthetic, ingest it, retain the chloroplast, and then become, for some brief period of time, photosynthetic themselves. So the next time you have a salad, I want you to imagine your arms turning green and then being able to go out on maybe a little bit sunnier of a day than today and get all the energy that you would need from photosynthesis. That's what these organisms are doing. And there are a wide array of them, including a couple members of the animal kingdom, the beautiful Elysia genus of sea slugs, and then there's also a flatworm. This one comes from coastal British Columbia that are capable of doing this. And then a number of protists, of single-celled eukaryotes that are capable of this as well. And this is a really profound acquisition of metabolism, right? We're talking about an organism that is genotypically heterotrophic, yet within its lifetime now is suddenly able to harness the energy of sunlight to get the organic material that it needs. And that's cool, right? I mean, these are the systems that got me into research as a scientist when I was an undergraduate, actually. So I have a deep love for them just because of their biological weirdness. But it's also really interesting from an evolutionary point of view, thinking about how these types of acquisitions allow metabolism to move throughout the tree of life. And I'm alluding here, of course, to the endosymbiosis hypothesis, which is our best guess at how eukaryotic cells got access to photosynthesis in the first place. So we think that photosynthesis was actually invented by prokaryotes, by the progenitors of our modern day cyanobacteria. And it was only when about 2 billion years ago, an early eukaryotic cell ingested and ultimately domesticated one of these free-living cyanobacteria that eukaryotic cells then got access to photosynthesis. And that was the origin of the chloroplast through this endosymbiosis event. Now this we term the primary endosymbiosis, where a free-living cell became an organelle, and it gave rise to two major lineages of algae, the greens and the reds, the greens going on to become our modern-day land plants. But in aquatic ecosystems, freshwater and marine, things got a lot more interesting because there have been a series of at least a half a dozen secondary endosymbiosis events by which other genotypically heterotrophic eukaryotes obtained one of these chloroplasts, permanently incorporated it into their metabolic machinery, and that gave rise to this huge extant diversity of eukaryotic phytoplankton that we have on the planet today, doing close to half of photosynthesis in aquatic ecosystems. Now, I would argue that if we want to understand the evolutionary origins of these eukaryotic phytoplankton and also how those origins will constrain their evolution in the future, more on that in a moment, our modern-day kleptoplastidic lineages that are currently acquiring photosynthesis give us insight into what some of the ecological selection pressures might have looked like to drive these lineages down this path from ancestrally heterotrophic to now photosynthetic. Now, if I want to position our modern day acquired phototrophs as hypothetical evolutionary intermediaries, I have to consider a couple of key evolutionary innovations. So the first of these is the evolved ability to retain a chloroplast in the first place. So you digest something, you ingest something photosynthetic, and then you retain that chloroplast and allow it to continue to photosynthesize. Now, I'm amalgamating a lot of cell biology steps in here, like interrupting the digestive process, tolerating the photooxidative stress, et cetera. But that's the key, right? You have to be able to hold on to this thing, at least transiently, to achieve some sort of fitness benefit from it. And then the second key innovation is the evolved ability to replicate the chloroplast and actually pass it on to your daughter cells, because it's only then, when you can vertically transmit the chloroplast, that you're a true eukaryotic phytoplankton. Now, again, we're confronted with a hypothesis that's hard to test with empirical data. So we turn to mathematical models and work led by Grace Casares, a former undergraduate researcher, and Dr. Alexandra Brown, a former postdoctoral scholar in our group. And Grace and Alexandra chose to work with an existing model of acquired photosynthesis that considers a phytoplankton that's growing photosynthetically and sharing the water column where it lives with a consumer cell. And when one of these consumers catches one of these phytoplankton prey cells and a predation event occurs, it retains or steals the chloroplast, represented by this green oval. Now, because this is a transient and not permanent acquisition of metabolism, daughter cells that are produced through photosynthetic growth of this consumer come out without a chloroplast of their own, and so they have to then go find and catch a prey cell to obtain a chloroplast. So we have this consumer then that's existing in these two states, out hunting, hungry, trying to get a chloroplast, or growing photosynthetically. Now, Grace and Alexandra modified this model in two ways to reflect the two key evolutionary innovations that we were just talking about. The first is that they allowed predation events to lead directly to heterotrophic growth of the consumer. And that gives us a single parameter, which we'll call the retention parameter, that can vary between 0 and 1, representing the proportion of predation events that either lead directly to the heterotrophic growth of the consumer. So if you never retain the chloroplast, you get your standard predator-prey model, to retaining it 100% of the time, in which case we recover our original model for acquired photosynthesis. Now, the second modification that they made was to allow plastids to be replicated during cell division of the consumer. And that gives us a replication parameter, and if that replication parameter evolves all the way to 1, that means that every time you divide, you pass on your chloroplast and you arrive at a model for the other evolutionary endpoint, our eukaryotic phytoplankton that can pass chloroplasts on to its daughter cells. So now we have a model with two parameters representing our two evolutionary innovations, and we can explore how lineages might travel through this evolutionary landscape. To do this, we use a branch of mathematics called adaptive dynamics. And the way that adaptive dynamics works is you start out with a population of cells. In this case, it makes sense to start out with those that represent our ancestral heterotroph that neither steals nor copies its chloroplasts. And then you ask, what happens if a cell in this population has a mutation that allows it to maybe hold on to a chloroplast that it's recently ingested? So the retention probability goes up a little bit. Will that mutant have a higher fitness? If so, will it take over the population? And we can repeat this mutation invasion algorithm many dozens of times in order to get evolutionary trajectories that look like this one, where this lineage has evolved through a period of time where it's behaving like an acquired phototroph and retaining chloroplasts from the food that it eats, and ultimately evolved a replication tendency of 1, which means that 100% of the time, like a true eukaryotic phytoplankton, it is reproducing the chloroplast when it divides and vertically transmitting that on to its daughter cell. Now, it turns out that for certain environmental conditions, the right availability of prey and of light, you can actually get two evolutionarily stable strategies that arise from this. First, shown here in blue, the solution that we saw in the previous slide, which is this phytoplankton-like permanent incorporation of this acquired machinery. And the second, shown in gold, is really a doubling down on kleptoplasty, on acquired metabolism. So these are lineages that have gotten really good at retaining a chloroplast 100% of the time if there's a predation event, but they don't bother trying to replicate it or vertically transmit it at all. So again, we have a prediction from a mathematical model. Can we validate this in any way? Is there any system out there in the real world that looks like this? And one of the cool things about working on acquired photosynthesis is that, in fact, there is. There's a really cool genus of ciliates, the Mesodynium genus, that are coastal ciliates. They, as ciliates, they propel themselves with a double band of cilia around their midsection. It's like a double hula skirt that they flip back and forth and go shooting through the water column. And within the same genus, we have member species that span an almost complete gradient of how much they rely on photosynthesis, from getting none of their carbon from photosynthesis to, in the case of the best described species, Mesodynium rubrum, getting more than 95% of the carbon in their energy budget from photosynthesis working with stolen chloroplasts. So they're really good at stealing this photosynthetic machinery. So good, in fact, that they can form red tides. So they can grow so abundantly that they can bloom and turn the water column red. This picture comes from the Columbia River estuary. You can see a bridge with a truck on it for scale to give you a sense of like how many square kilometers these guys can be the dominant primary producers over working with stolen photosynthetic machinery. Now, within the same genus, there's a sister species, Mesodynium chameleon, that gets about half of its energy from photosynthesis and half from directly digesting its prey. And if you're wondering, indeed, that chameleon species name is indeed reflective of the fact that we can get them to change color, so we can feed them different cryptophyte prey, the cryptophyte algae that they steal chloroplasts from, and when you feed them blue-green chloroplasts, they turn out green, like this guy up top. When you feed them red chloroplasts, they turn out red, and then here's one that's got a mouthful of each plastid type lit up like a little Christmas tree over there. If you want a better look at what the ultrastructure of these cells looks like, we've been experimenting with a relatively new microscopy technique called expansion microscopy. What you do is you fix a cell and you embed it in a gel, and then you hydrate the gel, which stretches everything to about 4 times normal size. And that means that suddenly you can image these cells using light microscopy instead of electron microscopy. Opens the door to all sorts of antibody fluorescent labels and stuff like that. But here, what I'm showing you on the upper left, the larger cell, that's our Mesodynium chameleon cell. And then these smaller cells are the cryptophyte algal prey. And I want to point out to you here in the middle, you can see this like yellow dotted structure here. That's one of these guys recently ingested, now in the process of being stripped of its organelles for use by the ciliate host. Okay, so we have someone who's almost entirely photosynthetic, someone in the middle, and then a third species within the genus that is entirely heterotrophic, so immediately digests the prey that it consumes. So there's this really cool natural gradient then that not only seems to fit with the evolutionary predictions of our model, but also allows us to test some additional hypotheses about what's actually happening within these cells as they evolve increasing reliance on photosynthesis. And so in collaboration with especially Erika Lasik-Nesselquist at the Wadsworth Institute, we've done a comparative transcriptomic approach to look at ciliate gene expression to ask what happens to the metabolism of these cells as they become increasingly reliant on acquired metabolism. Now, of course, one of the first things that we expect is that carbohydrate metabolism is going to look really different, right? You're plugging a little sugar generator into your metabolism, so surely that will change how you handle your carbohydrates. What I did not expect was the overall wholesale changes in metabolism that we see in these organisms as they become more and more photosynthetic. So what I'm showing you in the radar plot on the left here is the proportion of genes that are expressed throughout the genus that are expressed by any one of the particular Mesodynium lineages. So smaller envelopes mean fewer genes, larger envelopes mean more genes are expressed. And I want you to particularly notice here in the middle in red and orange in these smaller envelopes are highly photosynthetic Mesodynium rubrum lineages. So these lineages not only no longer do a lot of their own carbohydrate metabolism anymore, but they can't do things like make half of their essential amino acids. So here, open boxes, black boxes mean that we don't think the ciliate can make that amino acid for itself anymore. A filled colored box indicates that it does express the genes necessary to make that amino acid. Our highly photosynthetic lineages don't do their own fatty acid oxidation anymore. They don't even seem to produce peroxisomes. In general, as they become more and more photosynthetic, they appear to rely more and more on their prey to patch together their metabolism. And that's in stark contrast to their capacity for genetic information processing, for example, which is much more complete, indicating that these guys can copy their own DNA, but they can't make their own amino acids or their own cell membranes. So we're continuing to try to understand how plastid control evolves in this lineage and get a sense of the mechanistic cell biology that's going on inside of these cells, because we think not only is it really important to understand how chloroplasts came to be incorporated into eukaryotic cells, but because we think that will also give us a sense of some of the constraints on the incorporation of that plastid that evolved alongside those acquisitions. And this is of interest to us because not only do we then better understand the evolutionary origins of these eukaryotic phytoplankton, but we may also be better able to predict how they respond to future environmental changes. So knowledge of these historical acquisitions may help us to understand the capacity, for example, for adaptation to a changing climate. So I want to close my talk today with one more example of our work in the lab, considering how this acquisition history shapes evolutionary potential, particularly in what we call constitutive mixotrophs. So constitutive mixotrophs are mixotrophic because, like Mesodynium, they mix photosynthesis and heterotrophy within the same cell. But unlike Mesodynium, they have a permanently incorporated chloroplast. So they acquired this metabolism some time ago and permanently put it into their metabolic repertoire. Yet they retained the ancestral capacity for heterotrophy. So it turns out that most of our eukaryotic phytoplankton, except for the diatoms, actually are mixotrophic and still have the capacity to ingest some prey and digest them, which makes sense because that was their evolutionarily ancestral state. And because these organisms have two forms of metabolism within the same cell, they have two ways in which they can respond to, for example, rising temperatures. Because we know the metabolism responds to temperature. Generally, we expect that as temperatures increase, rates of metabolism accelerate exponentially until at some point it's too hot to do anything anymore. But it turns out that the acceleration of photosynthesis, shown here in green, is actually thought to be slower than the acceleration of aerobic respiration, which underlies heterotrophy, which is shown here in white. Now, if this is true, it means, as predicted by Suzanne Wilken about 12 years ago, that a mixotroph that's capable of doing both forms of metabolism should become more heterotrophic at warmer temperatures because that is the more metabolically favorable form of metabolism. And that's worrisome, right? Because then we're talking about an organism that, rather than pulling carbon dioxide out of the atmosphere through photosynthesis, is going to shift towards more remineralization and exhalation of carbon dioxide into the atmosphere through heterotrophy. Now, we think that the extent to which these mixotrophs are going to exhibit these responses is going to be impacted by their plasticity or their metabolic flexibility, which is constrained by how they incorporated that chloroplast in the first place. And will further be impacted by their evolutionary responses. These are organisms that are small, they have rapid generation times, large population sizes. They're exactly the types of things that we expect to mount a rapid evolutionary response to changing environmental conditions. So Michelle Lepori-Bouy, a former NSF graduate fellow, former master's student in our group, decided to explore this with an evolution experiment, in which she took mixotrophs and she grew them at their ancestral temperature of 24 degrees Celsius and then at colder and hotter temperatures for many hundreds of generations. And once you've given organisms a chance to evolve at novel environmental conditions, then you can do what we call a reciprocal transplant experiment, in which you take the cells evolving at the ancestral temperature, you move them to either the colder or hotter temperatures, and you compare those short-term plastic responses to the performance of the cultures that have been there for hundreds of generations evolving. Now, one of the first things that you check when you run an evolution experiment like this is to check for evidence of adaptation. So here on the x-axis, I'm showing you evolutionary time, and then on the y-axis, the difference in growth rates between the lineages that have been evolving at these temperatures to those that have been brought there relatively recently. So positive numbers are going to be evidence of adaptation. What Michelle found in both her cold and blue and hot and red evolved lineages of mixotrophs is that within fewer than 100 generations, they were able to grow faster at their new temperatures, so evidence of adaptation. What's underlying that adaptation metabolically? Well, it turns out that both of these evolved lineages at both cold and hot temperatures reduced their investments in photosynthesis, But in particular, here on the far right, in this red bar, you can see that the lineages evolving at hot temperatures are doing even more heterotrophy. They're eating even more than the ones that have not had the opportunity for that evolutionary response. So in a sense, evolution is doubling down on this metabolically favorable, this thermodynamically favorable form of metabolism, which means that evolution could potentially exacerbate this positive climate feedback loop, whereby warmer temperatures lead to more heterotrophy, lead to more atmospheric CO2, lead to further warming. And this is work that, this is supported not just by Michelle's experimental work, but also by mathematical modeling by Logan Gonzalez, who's a former undergraduate researcher and now a PhD student working on similar questions at Rutgers University. So that's a depressing thought to end a talk on, right? So I'm not going to end it here, because Michelle found this with one of the lineages that she experimentally evolved, but not the other. On the one hand, awesome, everything is not terrible. On the other hand, how then could we possibly predict which outcome will occur when, if it's, entirely random? So is there anything we can do to make more concrete predictions, ideally based on traits that we can measure about these mixotrophs today, that will help us better constrain these future predictions of responses to environmental change? So the hypothesis that we're currently working on to explore this is trying to link evolutionary responses to existing metabolic plasticity. So here, when I think about metabolic plasticity, if I think about low plasticity, I'm thinking about a mixotroph that has kind of a fixed ratio of chloroplasts to digestive vacuoles, and it's just chugging along doing the same thing regardless of the environment it finds itself in. But a highly metabolically plastic mixotroph is able to really adjust how much photosynthesis or how much heterotrophy it's doing in response to environmental conditions. There are a couple predictions out there in the literature about how plasticity should impact evolution. In our case, based on Michelle's preliminary data, we actually think evolutionary rates will decrease with plasticity, because that plasticity helps buffer natural selection. So basically, you're doing okay just working with the metabolic flexibility that you have, and that reduces the strength of selection, slowing down rates of evolution. So to test this hypothesis, we need to measure plasticity across a bunch of mixotrophs, and we need to measure rates of evolution across a bunch of mixotrophs. Fortunately, we had some fantastic undergraduate researchers who have made a phenomenal start to this. So Gina Barbalia, one of our undergraduate researchers, characterized the metabolic plasticity of 8 different mixotrophs from the same genus as those Michelle experimentally evolved previously, by basically putting them in a gradient of environments that push them from high light to low light and high prey to low prey, to basically say, how do you respond to this buffet of different resource availabilities? And that allowed Gina to build essentially these plasticity envelopes, the details of which are not important except to point out to you that the X and Y axes are different scales, meaning that some of these mixotrophs have more metabolic flexibility and more plasticity than others. And then building on Gina's data, another undergraduate researcher in our lab, Jessalyn Lee, conducted a massive evolution experiment where she took those 8 same lineages and she evolved them at both cold and hot temperatures, and literally went into the lab every day and counted them so that we could get really high resolution measures of evolutionary rates, which, like, woof, that's a lot of data. Okay, so from Jessalyn's data, then, we can estimate growth rates. I don't expect you to get much out of this plot except being impressed by how many data points she actually collected. We can measure the growth rates over evolutionary time. And in particular, we can look at how fast those growth rates change over evolutionary time to get a sense of how speedy evolution is. And then using those data, we can now fill in our metabolic plasticity from Gina's data and our evolutionary rates from Jessalyn's data and test our hypothesis. And here's what it looks like at hot temperatures. In particular, the flexibility of these organisms to make photosynthetic investments seems to be predictive of evolutionary rates in the direction that we expect. But because biology is hard and complicated, only at hot temperatures. At cold temperatures, we get the opposite trend, and it's much more marginally statistically significant. So we're still scratching our heads trying to figure out exactly what some of the subtleties here are. I think it may have to do with the extent to which these organisms already have pre-existing mechanisms of tolerating cold temperatures, but not hot temperatures. But I can talk more about that another time. But overall, right, what this does is point to the importance of understanding these acquisition histories and how they shape the contemporary ecology of these organisms by allowing them to extend their metabolic niches, but also shape their evolutionary futures by constraining how plastic they are and therefore how responsive they are to environmental change in the future. So, in sum, today we've looked at a couple different examples of acquired metabolism to understand both ecological and evolutionary impacts. In our study of tree ectomycorrhizal mutualisms, we were exploring how swapping out different fungal partners or investing in a portfolio of fungal partners basically lets the tree wear different metabolic hats depending on its local environmental needs. And then we took a look at acquisitions at the subcellular scale, thinking about what we can learn from studying organisms that steal chloroplasts and how those acquisitions can really allow metabolism to jump across the tips of the branches of the tree of life. And finally, we thought about what the downstream consequences of those acquisitions, even when they become permanent, are for the capacity of these organisms for future evolutionary change. So with that, I want to thank you all so much for your time and attention and the wonderful visit to Bodega, and I'm happy to take whatever questions we have time for. Thank you.
Thank you. Oh, that was really cool. In the model, looking at how it switched between the two different stable states, what were sort of the factors that would cause it to switch? And was it mostly temperature, or is it other things too that are causing these two strategies to be good for the different organisms?
Yeah, thanks, Claire, for that question. You're talking about the kleptoplasty model? Yeah, so question about why we observe these two evolutionarily stable strategies, emerging. And it turns out that those evolutionarily stable strategies coexist in the same environmental conditions. They have to be intermediate light levels because there has to be some selective advantage to stealing the chloroplast at all. But what really governs that is that there's a trade-off, which is based on empirical data in the mesodynium system, between how good you are at stealing a chloroplast and how good you are at eating. So in the mesodynium system, as these lineages become more and more photosynthetic, they also become more and more specialized on the prey that they consume, which makes sense because you've got to match the organelles and apparently patch up your entire metabolism by finding just the right source of organelles. So they become very specific in the types of prey that they feed on. And so the reason we're seeing these two evolutionarily stable strategies is that in one case, these lineages have, to anthropomorphize a little bit, chosen to get really good at stealing the organelles, and in the other case, they've chosen to retain the capacity to eat a wider diversity of prey.
That was really cool. I have a question about some of the mycorrhizal fungi, and I'm wondering what the environmental filtering process for the mycorrhizal fungi that are available in the environment. If there's a really variable environment, is the pool of fungi available constant, or if you have sort of a shift to one, so environment A versus environment B, whether that changes the availability of potential symbionts as well?
Yeah, thank you. Such a great question about the extent to which environmental filtering is playing a role in the ectomycorrhizal system. So that's certainly something we think about a lot in that Mendocino chronosequence, right? Are we seeing high resource transport fungi on these older soils because the trees are picking them, because those fungi are more competitive, because the environment is filtering for them in some way? Our master's student, Jasmine Sedano, is exploring that now with some experimental studies in the greenhouse to try to disentangle some of those coinciding factors in the field. But broadly, I would say that we know that some fungi are more in absent association with the trees. These fungi are persisting in the environment as spores. And some fungi are able to persist for longer periods of time and able to colonize successfully from spores better than others. So one of the biggest gradients that we see is the presence or absence of host trees. Because if you're missing host trees, then you lose a fraction of the fungal community that's not able to viably colonize from spore. And so that's important when we consider like reforestation of fire, after fire, et cetera. It's something that's thought about a lot in the timber industry and leaving behind some trees in clear cuts so that there's someone there nurturing the fungi so that they're available as colonists when you reseed or replant those locations. So I think it's a great question. There's a lot to continue to explore there. And at least some of that is going to be filtered by the presence of host trees to maintain that carbon-requiring fungal community.
Hello. I had a very similar question, so I'll ask a different question. I was curious, in your mesodynium plots that you were showing, there was a temperate and a polar of the same species. I forget which one. One on either end of the spectrum.
Yes.
I was just curious if you could talk about some of the differences you saw between those regional regional differences of the same species, and if it either supports or conflicts or gives you cool ideas for some of the temperature work you're doing.
Yeah, thanks. That's such a great question. So our highly photosynthetic Mesodynium rubrum species, actually the one that I worked on for my undergrad thesis is the polar one. It's from Antarctica, from McMurdo Sound. Been in culture for, well, now I have to add on the 20 years that it's been since my undergrad work, for probably close to 40 years now. And the temperate one is actually, this one I believe is from Spain. We have one from Chesapeake Bay. There's a few different examples of that. You know, to our surprise, despite the biogeographic distances between these lineages, they were much more similar to one another than they were to other species in terms of their gene losses. There's a challenge in working with these ciliates in annotation. Anyone who's worked with protists will feel our pain there and our ability to actually accurately identify the genes. So we're only annotating about 50% of the genome, and it's possible that some of, especially like cell membranes and membrane fluidity, some of these secondary modifications that are happening to the lipids that we think must be encoded in the mesodynium because they're not encoded in the prey and they're different lipids from what the prey have. We're just not able to accurately identify yet. But so I completely agree. I think that's a great idea, and we're just not quite there yet in disentangling what we would expect to be some of the low-hanging fruit of comparing polar and temperate strains. Great.
Yes.
Or I was very unclear.
No, it was a great seminar. Thank you
Thank
very
you.
much. I have more questions about the mycorrhizal associations. So how does let's say the specificity of some of these interactions versus some trees that have associations with many mushrooms and others where it's more specific, how does that play into this?
I think that's a great question, and those of you who know something about Ectomycorrhizae will not be surprised to know that I do a lot of my work in Douglas fir, which is known to have a particularly high diversity of fungal associates. I don't think we have the data at the moment to differentiate between sort of like evolutionary accidents and evolutionary constraints that versus like environmental factors that affect how many fungal associates you have. So to make that a little bit more explicit, I don't know that I could confidently stand here in front of you today and say that ectomycorrhizal trees that have fewer fungal associates have narrower, more climatically stable ranges. But that's a hypothesis that might arise from this work, right? That Douglas fir with its diverse associates is also going to have more broad tolerance of different environmental conditions. The reality is that I think we need to do a lot more characterization of the fungal traits and environmental factors that these fungi can tolerate to accurately characterize that. but absolutely in more specialized plant fungal associations, we'd expect to see less of this bet hedging phenomenon simply because there are fewer fungi to work with. And we also expect, and this is something that Laura is doing really cool work on, perhaps more refined partner recognition mechanisms in these more specialized associations compared to Douglas fir, which is more general in its associations.
I'm going to take this chance to sort of get a couple of Zoom questions that I'm going to smush together, all related to the mycorrhizal fungal question. So I'm going to combine two here. So for a tree with potential ectomycorrhizal fungi, how does it, quote, decide/learn what fungi to build a partnership with? And the related question is, do trees in more variable environments tend to have a more diverse mycorrhizal assemblage?
Yeah, great questions about the patterns that we see, both in terms of the mechanisms by which trees are choosing, quote unquote, choosing partners, and also what patterns we might expect to see in the field. So Laura Bogar has done, I think, some of the best work in the world on quantifying investment strategies in trees by using mesocosm experiments to manipulate the quality of fungal partners and actually looking and watching the trees preferentially allocate their carbon below ground to the side of the root system that has the highest quality partners. So that's some of our evidence of what choosing might look like in at least a post-association form. Pre-association, I think, we know a little bit about some of the chemical language that trees and fungi are using to communicate with one another and say, hey, it's a viable partnership, you can get on my root system, it's fine. But there's a lot of open questions there about how that's being done. And honestly, to me, these experiments looking at preferential allocation of resources, the fact that we never, in ectomycorrhizal systems or arbuscular mycorrhizal systems, you never see the low quality fungus be completely cut off is, in my opinion, one of the more compelling pieces of evidence for the bet hedging hypothesis, right? That these associations are imperfect and that there is some leakiness. And that may be an evolutionary accident or an arms race thing. It might also be an evolutionarily beneficial type of mistake to make. The second question was about the diversity that we see across gradients and environmental variation in the field. We have an NSF project trying to test the bet hedging hypothesis, but particularly trying to test that specific hypothesis about whether we see a higher diversity of fungi in more variable environments in the field. The answer, based on our field data, is there's not a clear signal. I find it difficult, which makes me sad because I want to support the hypothesis, but I find it difficult to reject the hypothesis because these are all secondary growth, so locations that were previously cut down and then reforested. There's probably also some community assembly processes that are happening as well, and so we see a signal of fungal community composition and environmental variation and tree performance, but not necessarily that diversity environmental variability signal that we might immediately hypothesize. But I think there are a lot of other systems and other locations in the world and more ideal field settings that we can look for that. So I'm not ready to rule out the bet hedging hypothesis based on those data yet.
My question's on. Yeah, thanks. My question's also related to the bet hedging hypothesis about how more variable environments should favor a more diverse portfolio. And I'm just wondering, it seems like all environments are variable in some way or another. So for example, in your AmNat model, can you say a bit more about what type of temporal variability is important? Is it the frequency of change or the magnitude of change, or what are you looking at there?
Yeah, thanks so much for that question about the types of variability that we expect to see as the most important. The paper suggests that there's essentially a threshold that relates to, it's about related rates, right? So if I stop investing in this fungus, how quickly will it die back from my root system before the environment changes and I need it again? And if that rate is too fast, then I better pay that fungus carbon even when it's not the environment in which I need it in order to maintain it as available to me when the environment changes. And so the answer to your question about what's the most important, is it the frequency of change or the magnitude of the difference in the quality between the fungi and the different environments? Absolutely, the frequency of environmental change is super important because that's going to control that time scale of the mortality of the fungus. But it also matters the way that those fungi differ from one another. So for example, if the fungi differ in their mortality rates in the ideal versus non-ideal environments, then it's about both the difference in the magnitude of the difference between the fungi and the environments that they're experiencing, and also the frequency with which environments change. If it's about the yield, like the per carbon nitrogen return from the fungi, then it's much more about the frequency of environmental flipping back and forth.
Okay, yeah, sorry, I know everyone has so many questions because this stuff is just so cool. But maybe this is a silly question to end with, but I am curious about, so there's this stealing of chloroplasts that happens. I know there's other useful organelles that might be stolen for purposes, and I'm just wondering if that is something, like, I don't know if there are examples where like mitochondria are taken to be like an extra heterotroph. I don't know. Yeah, curious about that.
Yeah, such a great question about the extent and diversity of organelle stealing in biological systems. And I love protists because, whatever weird thing there is to do with in the world, they will figure out how to do it. Yes, absolutely. There are examples of organisms that steal mitochondria. Actually, I just learned this last year at one of our molecular biology seminars. Apparently, cancer cells steal mitochondria. Yeah, so that's cool. I guess I should give animals some credit for doing interesting things. So there are examples of mitochondria being stolen. We see this actually sometimes in organisms that live in low oxygen environments, that there are mitochondria that appear to be better able to tolerate lower oxygen environments, and so stealing those may help you to tolerate that low oxygen environment yourself. Mesodynium itself is actually stealing chloroplasts and mitochondria. And I didn't tell you this, but they're also stealing functional nuclei. So they steal the car and they steal the owner's manual to the car, and then they repair the car with the owner's manual. Yeah, so I think that's part of why they can surrender so much of their metabolism and pseudogenize so many of the genes related to core metabolism, is because they're literally taking someone else's genetic instructions to plug that gap. So they function a little bit more like an exoparasite, if you will, where they're stripping down and simplifying away their metabolism and allowing someone else to just kind of run the show in this very controlled way. So great question. Lots of examples of acquired metabolism, not just through the theft of chloroplasts, but of other organelles as well.
Great. Thanks, everybody, for the great questions. And for all the people on Zoom, let's thank Holly one more time for a doggone good talk.